Good afternoon, everybody. My name is Sylvie Garneau Tsodikova, and today I'm here to tell you about new drugs for currently untreatable fungal infections. And you might say, Sylvie, fungal infections are not a problem at all. Well, that's where you would be wrong. Fungal infections are actually, could be deadly, and we're currently in dire need for new treatments. You might have seen headlines in the New York Times in a few years ago, or even recently, such as a mysterious infection spanning the globe in a climate of secrecy. Well, fungal infections are by no means secret, but they can be deadly. And you might have seen other headlines such as to fight deadly Candida auris, New York State proposes new tactics. Candida auris is one of these fungal pathogens against which most, if not all, of our current antifungals do not work. I have a student that just started to work at the New York Health State Department to try to fight this Candida auris epidemic. And then came COVID. And you might say, COVID is a virus. What does that have to do with fungal infection? Well, you might have seen headlines such as how COVID created a perfect storm for a deadly infection in India, fungal infection, or headlines that are closer to home here in the US, like Candida auris outbreak in a COVID-19 specialty care unit in Florida. So you can see that fungal infections can be deadly. If I have not convinced you yet, you might still be thinking about superficial fungal infections, like those that affect your hair, nail, and skin. This is not what we're excited about. What we're excited about and focusing on are systemic fungal infections, those that are lethal and that will affect your brain, lungs, heart, liver, spleen, and kidney. These affect millions of individuals worldwide every year. And fun fact, actually not fun, but a fact, more people die from the top 10 invasive fungal diseases than from tuberculosis or malaria. So let that sink in for a minute. More people die from top 10 fungal infection, the invasive one, than from TB or malaria. If that's not enough to convince you that we need to work on antifungal drug development, let's look at staggering mortality rate from systemic fungal infection worldwide. If you look at diseases such as aspergillosis, candidiasis, cryptococcus, mucormycosis, or pneumocystis, you will see that they infect hundreds of thousands, and in some cases, more than millions of people everywhere in the world every year. And amongst this infected population, mortality rate can go from 20, and in some cases, up to 95%. 95% of these people will die from these fungal infections. Now you might say, well, we do have antifungal agents on the market, and that would be true. But there are problems associated with our current antifungals. There's toxicity problem, and a lot of the fungal pathogens have developed resistance to all of the antifungal agents that we have, or many of them. You can see here examples I told you about Candida auris a few minutes ago. Candida auris, a drug-resistant germ that is spreading in our healthcare facilities. So these fungal infections are spreading in our hospitals, and we need new antifungal agents. But don't worry, we have a solution. We have novel classes of antifungal agents that could have new mechanism of actions different than the ones that are currently on the market. And I'm showing you one category here, and we're calling them the HA compounds. There's a big name associated with them. Don't worry about it. Just remember HA, like ha-ha moment. And we have patented these compounds in 2019, so there's plenty of time left on the patent. And if you look at their structures, you will see that they're very simple. They're comprised of some aromatic rings, these 6-membered rings, that can be linked directly together, or there could be a linker linking these two aromatic rings, and we could change the aromatic rings themselves. We can also easily modify what we attach to these rings. So you can see that these simple molecules can increase in structural diversity very rapidly. Now, a beauty of these compounds is that they are easy to synthesize. A few high-yielding synthetic steps will lead to these molecules. We currently have 7 families that we're working with. We can expand to more families if we change the linkers. And these families, we have a lot of compounds that are in these families right now. I'm going to show you a table with a lot of data. Don't worry about the numbers. These are minimum inhibitory concentration values. What you need to know is the lower the number, the better. You don't even have to look at the numbers because I color-coded it for you. Red means bad, not good antifungal activity. Yellow means good, and green means excellent. So you want more green. And I'm showing you just a few results here. And you can see that our compounds are broad spectrum. They have activity against Candida species, including this deadly Candida auris, Aspergillus, and Cryptococcus. And they have high potency as high as 0.06 micrograms per mil. So that's all good. One thing that I haven't put on my slide is that some of these compounds will prevent biofilm formation or will destroy preformed biofilm, which are matrices of these antifungal agents. Now, one thing that we need, one of the problems we want to alleviate is the resistance problem. So we need to see if our compounds develop, if the fungal strain develop resistance to our compound, and they do not. I'm just showing you an example here in the bottom right corner, where you can see that after 13 serial passages, there was no resistance developed by the fungal strain to our compound. So that is also exciting. Now, you need to think when you want to treat patient, you want to make sure that your compounds are not toxic to mammalian cell, because that would be bad. So I'm showing you here 2 mammalian cell lines, and you can see that the mammalian cell lines are surviving really well at high concentration, much, much higher than the concentration that we need to kill the fungal cell. So that is really good. Our compounds are not cytotoxic. We also need to think when we develop antifungal agents, if our compound will affect the blood of the patient, because we don't want to destroy their red blood cells. That would be bad. So you can see here that at high concentrations, our compounds are not hemolytic or have very low hemolytic activity. So our compounds do not destroy red blood cells. That's a plus. You also need to think not only about the blood of the patient, but about the cardiotoxicity. So you want to not have hERG liability. So what we want to see here are compounds that have either IC50s in an acceptable range, which would be between 1 and 10 micromolar, or that have very low affinity for the hERG, which is more than 10 micromolar. And I'm showing you some examples here where we can see that the IC50 values are more than 30 micromolar, which is awesome, and some that are in the acceptable range. So here you see that our compounds are also not cardiotoxic. So it seems like our compounds have high potential. So what are we doing next? What are we trying to do now? We've eliminated some of the current target for antifungal agents, and now we are trying to find what is the target for our compound, and we will need to validate that. We have a few methods that we're using to try to find the target. We're further optimizing for their properties, optimizing the compounds, and we need to do preliminary pharmacokinetics, metabolism, as well as bioavailability studies. Now, there's potential in market. They could have all the properties to reach IND status, and there's opportunity for accelerated FDA review to regain AC and LPAD pathway. If we look at market analysis and competitive landscape, the global market size for antifungal drugs in 2016 was at $12.5 billion, and with a compound annual growth rate of 1.5%, is estimated to grow to $17.2 billion by 2025. Now, we have competitors, which is awesome because that means that our compound, what we're trying to do is important. For example, Amplyx Pharmaceuticals was acquired by Pfizer in 2021, April of this year. So that is really exciting because the compounds of these pharmaceutical companies or these startups are different in structure, completely different in structure, and they have mechanism of actions that are probably not or are not the ones that our compounds have. So we have competitors, but our technologies is very complementary and different. Okay, so we want to start a company that's called Microbics. Potential funding sources could include NIH Kinetic Grants, SBIR, Department of Defense, and of course we're looking for investors. So this is why I'm so thankful for the Equalize opportunity to be pitching to you today and having had my mentor at ArchVenture. We've had funding so far, and I'm really thankful for the funding that we've had to be able to reach the point that we are at now. So who are we? We are a highly collaborative team that has for mission and passion to fight infectious disease health threat. As I said at the beginning of the presentation, my name is Sylvie Garneau Tsodikova. I am a professor of pharmaceutical sciences as well as associate vice president for research at the University of Kentucky. My expertise is in medicinal chemistry, microbiology, molecular biology, and biochemistry. And I am supervising and managing this project along with Professor David Watt, who brings an additional expertise to the team, which is to have brought product to market by starting different companies such as Epionc and ELISA Technology. one of the main scientists working on this project, on this antifungal, is Nishat Tamban Chandrika, who is just an amazing researcher. And I will leave you with one parting thought. They say an apple a day keeps the doctor away. And I hope that one day, if you have a fungal infection, you'll be able to say, ha, Sylvie's ha compound will be able to keep my fungal infection away. Thank you for your attention.
That was terrific. Thank you so much, Sylvie. For our first question, Chris, would you like to go?
Sure. Yeah, really nice presentation. I think you became a good advocate for systemic fungal infections. So just in terms of the seed, I presume you're looking for a seed to optimize some of these hits that you have. And if you can estimate how much money and how much time to get to a viable HA lead. And then the other one is, can you just talk about the market dynamics a little bit and how this may or may not be similar to antibiotics, or how it may be different, where there haven't been a lot of successful. I used to market Ambisome, amphotericin B, and still many of the drugs for resistant are still held back. And so they try everything else first before they go. So maybe a little bit about the market dynamics and just how much you think you need to get to a lead.
Yeah, so we will need, in terms of what we want to do next, I think it is important to put these compounds in animals to figure out their metabolic stability and figure out the PK/PD and all of these things. So I think that's one of the first things we want to do. And we know that animals are more expensive than in liquid culture. So probably I would say, I'll say 5 million just to get started with all these animal studies. And I think it's going to be important to also, we talk about systemic fungal infection, but these compounds could also, and I put Department of Defense there, could also be useful if there's infection in wounds and things like that. So some different type of animals, just not my studies, would be useful. So I would say probably like 5 million over there. And how long will it take? I would say probably a few years to get the preliminary important data. Now we need to optimize the compounds. There's probably going to be some solubility and some metabolic problems. I know that one of the main problem will be the CYP3A4, for example. That could be a main problem with the azoles. I don't think this is going to be a problem here. We have looked, some of these compounds are okay in terms of solubility, some of them are not, so we'll need to work on that. In terms of problems like antibiotics, I would say, I think people are waking up, and maybe that's a weird way of saying it, but we are, a few years ago when I was young, when I started my career, people were telling me that antibiotics and antifungals were a dead end and nobody needed them. And people are finally realizing that they're important. I'm encouraged by the fact that APX001 was acquired by Pfizer. So I have a lot of hope for these compounds.
No, that's good, Sylvie. I want to leave the other judges a chance to ask questions as well. Yeah.
Great, thank you.
I talked too much.
I just also wanted to know, but just the timeline of all this. You talked about what you need and how much, but just in terms of the timing of all it, when do you expect to kind of get to your next inflection point?
Yeah, I think I would say I would go by slices of 2 years. Like it's going to take more than 2 years, of course, except if we get a lot of money and a lot of people to help work on this, but because that takes a lot more time. But I think in slices of 2 years to get the animals, all the animals data that we need. And I think having a target will also be very important. So we're trying, I told you there's not much development of resistance against our compounds. So we're trying to develop resistant mutants so we can figure out the target. We also have different probes to try to pull down the target that we're using. And we have libraries of fungals that have mutated targets. So we're trying in that way as well. So we have 3 approaches to try to identify the target. And once we have the target, hopefully it's going to be a completely different target than the other compounds, which would be amazing because then resistance would be even harder. Well, the fungal strains would not be as used to see these types of compounds attacking them. So I think if like getting a target is also in parallel with the animal study are the two main milestones in the next few years that I would like to accomplish.
Thanks.
Christine, do you have a really quick one?
Just very quick. Have you done on your MICs, have you done clinical isolates, or were those lines? And then, have you compared against the competitors that are in the clinic?
Yeah, so we have some ATCC strains, but we also have a variety of clinical isolates. And I just put a few examples here, but we also have like the mucormycosis. We have like, I didn't put everything, but we have clinical isolates of a variety of fungal strains. And they are resistant to the current antifungal that we're testing, but they're not resistant to our compounds. So that's important because we don't want to just use something that's susceptible to everything. So yeah, we're looking at resistant clinical isolates for sure.
Great. Thank you. Thank you,
Thank you
judges, for your questions.
opportunity.
Yeah, thank you, Sylvie, for a terrific presentation.